In this section, we'll discuss about different levels of resolution of HLA alleles that can be achieved by molecular typing during identification of these alleles. In this example, we have identified all the details of HLA allele, and this is called allele level resolution. So if we know all the details of HLA allele, then we have achieved allele level resolution of that allele. And if we only know first two digits or first field of allele, that is called low resolution. That only tells us allele group of an allele. In this example, we have an allele and we know this allele at 2-digit or low resolution level, and it only tells us that this allele belongs to HLA A2 group. However, there are several alleles in HLA A2 group, and at low resolution we cannot tell which exact HLA A2 allele it is. In this example, we have 6 alleles of HLA A2 group, and on low resolution we can see that all these alleles belong to HLA A2 group, but we do not know whether all these alleles are similar alleles or different alleles. On further analysis, we can see that all these alleles are actually different alleles, and these alleles will result in HLA molecules that will have some difference in their amino acid sequence, or more precisely, some difference in their amino acid sequence of peptide binding region. So if we know an allele at 4-digit level, that will tell us which exact HLA molecule that allele will result in, and that is called high level resolution. So if we know an allele at high level resolution, we know which exact HLA molecule that allele will result in. So in this case, we can see 6 different HLA alleles of HLA A2 group will result in 6 different types of HLA molecules. If we try to identify these HLA molecules by serology with help of HLA antibodies, HLA A2 antibody will react or cross-react with all these 6 HLA molecules, and all these molecules will appear as A2 antigen. And with help of serology, we cannot identify which exact HLA molecule it is. And if we want to further identify HLA molecule, then we need to perform structure analysis of individual HLA molecule to know structure of HLA protein. So with help of serology, we cannot identify which exact HLA molecules a person has, as many HLA molecules appear as one antigen. However, in practice, we can identify HLA alleles at high resolution level, and that will tell us which HLA molecules a person has. So if we want to identify which exact HLA molecules a person has, then we identify HLA alleles at high resolution, and that will tell us which exact HLA molecules a person has. For solid organ transplant, identification of HLA alleles at low resolution or antigen level typing is generally considered enough. However, for bone marrow transplant, identification of HLA alleles at high resolution is considered appropriate. So, low resolution typing of allele by molecular method tells us allele group of that allele. And that allele can be one of several alleles of that allele group. And low resolution typing generally corresponds to antigen level typing. Like if we find an allele of A2 group on low resolution, it can be one of several alleles of HLA-A2 group. Similarly, if we find A2 antigen by serology, it means it is one of several proteins of HLA-A2 group. So these 6 alleles belong to HLA-A2 group on low resolution, and we do not know which exact allele these are. And all these HLA-A2 alleles will appear as A2 antigen by serology. So HLA molecules of these alleles will appear as A2 antigen on serology. And we will not be able to identify which exact HLA molecule it is. So low resolution level typing generally correspond to antigen level typing. So molecules derived from HLA-A2 allele group will appear as A2 antigen, while molecules derived from HLA-A1 group will appear as A1 antigen. And if we want to know exact HLA molecules, then we will need to perform further analysis and identify alleles at high resolution to identify exact HLA molecules. In this example, we have 6 HLA alleles that appear similar on high resolution, and these HLA alleles will result in HLA molecules that will have similar structure of peptide binding region. However, on further analysis, we can see that these alleles are actually different alleles. However, all these 6 alleles give rise to HLA molecules that have similar structure of peptide binding region. So different HLA alleles can result in HLA molecules that have similar structure of peptide binding region. So if a donor and recipient have HLA alleles that are similar on high resolution, they will have HLA molecules that will have similar peptide binding region. However, on further analysis, they may have same alleles or they may have different alleles. Another level of resolution of HLA alleles is intermediate resolution. At this resolution, we know allele group of HLA allele, and we know this allele is one of given few alleles. In first example of intermediate resolution of allele, we can see that this allele belongs to HLA-A2 group, and this allele can be HLA-A21, A22, A27 or A220. So this allele is one of these 4 alleles. And if we want to know exact details of this allele, then we will need to perform further analysis.